On behalf of Cambridge Health Tech Institute's Global Web Symposia Series and our sponsor, Integral Molecular, I'd like to welcome you to comprehensive specificity profiling of antibody-based therapeutics using the membrane proteome array. My name is Elizabeth Lamb, and I'm the host and moderator for today's webinar. Now I would like to introduce our presenters for today. First is Rachel Fong, Senior Project Leader, Alliance Management, Integral Molecular. Our second presenter is Timothy McLaughlin, PhD, DABT, Executive Director, PCS, TA Head, Cardiovascular and Metabolism, Novartis Institutes for Biomedical Research. And our third presenter is Dmitry Dimitrov, PhD, Doctor of Science, EVP and CSO for Abound Bio. And with that, I will turn the webinar over to our presenters.
Thank you so much, Elizabeth. Hello, everyone, and thank you for joining our webinar today. My name is Rachel Fong, and I'm the Alliance Manager at Integral Molecular. Today, I'll be speaking to you about the membrane proteome array, the technology, its application, and benefits. Here's a very brief outline of what I will cover in my portion of the webinar today. So first, I'd like to give you a very brief introduction to Integral Molecular so you can learn about who we are and what we do. I will cover why specificity testing is important, and then I will focus on the membrane proteome array technology itself. So I'll talk about what the technology is, the features and benefits, the use of MPA data in regulatory filing, and also show a couple of case studies. And then to finish up my portion, I will talk a little bit about what a standard MPA project looks like so that you know what to expect if you decide to work with us. Integral Molecular is a biotechnology company located in the city of Philadelphia in the United States. We have been around for 20 years, and we are the industry leader in discovering antibodies against membrane proteins. So in our time in the business, we have developed a number of different technologies for discovering and characterizing antibodies against membrane proteins. So most commonly, we're working with GPCRs, transporters, ion channels, immuno-oncology targets, and then we also have quite an extensive virology arm of a company, which in the last year with the pandemic, we've really pivoted to offering a lot of technologies for researchers who are working on therapeutics against SARS-CoV-2. Throughout the years, Integral Molecular has worked with over 400 companies, including the top-tier pharmaceutical companies and many well-known research groups all around the world. They trust us, and as you can see from the slide here, we do have our work shown in over 250 publications and patents, most of them from our customers, including publications in the top-tier journals Cell Science and Nature. So I think this really goes to show the validation of our work and the quality of the science that we do here. Before I do the deep dive into the membrane proteome array, I do want to briefly mention the other technologies that we offer here at Integral Molecular. First is the MPS antibody discovery platform, which is a full soup to nuts service for antibody discovery, specifically for antibodies against membrane proteins. Next, we offer our lipoparticles. These are virus-like particles that have a high concentration of membrane proteins on their surface, all presented in native conformation. Lipoparticles have many different applications, a really great tool for anyone working with membrane proteins, and can be used for immunizations, panning, ELISAs, and biosensors, among other applications. Next, we also offer shock immunogenesis epitope mapping services. Our epitope mapping results in very high resolution epitopes, even for conformational antibodies on complex proteins. So if you'd like to learn more about any of these other technologies, feel free to check out our website or get in touch with us after the webinar. But for today, for the webinar, we're really going to focus on the membrane proteome array service. Before I get into the technology itself, I want to talk a little bit about the general idea of specificity screening in drug discovery. Why is specificity testing important? We all know the huge amount of resources required to bring a drug to market, both in financial terms and the amount of time required. Since it is such an investment, more and more companies are thinking about ways to de-risk their programs earlier before clinical development so that poor or non-specific candidates can be replaced or engineered before too much time and or money is invested in them. Specificity testing is one thing you can do to help decrease the risk of drug failure. Even though specificity testing is required for IND, we do recommend testing drug candidates in the late discovery or early preclinical stage to give yourself as much leeway as possible. In our experience, we have seen that about a quarter of the antibodies tested at integromolecular alone exhibit polyspecificity. And this polyspecificity is most commonly to proteins that are completely unrelated to the intended target. Knowing this information earlier in your drug development process allows you to make informed decisions about which candidates to further progress into preclinical studies. Because moving forward with an antibody that is also binding an off-target protein can have unpleasant consequences. What that polyspecificity or binding to an off-target protein means is that an antibody could have increased risk of failing in clinical trials due to severe adverse events, or it could be approved for the market but suffer major side effects or be marketed with a black box warning, as has been the case for previous antibodies that were withdrawn from clinical trials or are currently on the market. So to have the best chances of avoiding these situations, Integral Molecular developed the membrane protein array platform, which provides a comprehensive specificity assessment that will reduce risk for off-target liabilities in clinical studies and beyond. The membrane protein array, or MPA for short, is a fairly straightforward concept. We have 6,000 full-length human membrane proteins arrayed in 3D4 well plates, one protein per well. We transfect these proteins into human cells, and then we can take the antibody of interest and screen it across the MPA by flow cytometry to measure binding. What we come out with is a specificity profile of that antibody. So you expect to see binding to the known target, and then we are then assessing to see if there is binding to any off-target proteins. In terms of what we have in the array, there are 6,000 total proteins, of which 5,300 are unique. That covers about 94% of the human membrane proteome, and we do have very good coverage of all different types of proteins, including single-pass proteins and multi-pass GPCRs, and also ion channels and transporters. All of our proteins are expressed in unfixed human cells. This is very important for maintaining native epitopes, and I'll discuss this a little bit more in a following slide. But this also means that the MPA can lend itself well to functional screenings. Here is a short case study of some work that we did with Don Siegel's lab at Penn Medicine a few years ago, but was recently published this year. What we did for them is that we tested two of their antibodies, P410 and P46, on the membrane protein array. These were antibodies developed against GFR-alpha 4 involved in thyroid cancer. And you can see here on the left-hand side, P410 Fab bound GFR-alpha 4, looks very specific to that target. But on the other hand, P46 Fab, we identified binding to GF4 alpha 4, but also binding to SYT2, which was considered an off-target hit. Around the same time, Don's lab was also performing some in vivo studies, and what they saw was they identified the antibody in the thyroid, which is obviously where it was expected to bind, but they also saw the antibody in the adrenal gland. Putting our two pieces of data together, we saw that SYT2 is actually a protein that's only expressed in the adrenal gland. So our MPA was able to identify this very physiologically relevant off-target binding that was also occurring in vivo. This information was obviously very helpful to the group, and they were really able to make better decisions and prioritize which leads they wanted to move forward with. Now I want to shift over to speaking more about the features and benefits of the membrane proteome array. So I mentioned earlier that we do use a flow cytometry-based platform to perform these screens. So really, we are compatible with all different biotherapeutic modalities as long as there's a way that we can detect it by flow cytometry. So standard IgGs are very common for us to work with, but we have also successfully screened scFvs, bispecific antibodies, nanobodies, as well as peptides. If you are working with a more unique or proprietary type of modality, we're always happy to speak with you and figure out a way that we can screen your molecule in our platform. CAR-T is, of course, a therapeutic modality that is becoming more and more popular. We have worked with a number of different CAR-T companies throughout the years, and we are the only platform that can screen CAR-T modalities in 3 different ways. First is a more standard binding assay using the receptor scFv portion of the CAR-T, but we can also perform cell binding and functional assays on the MPA to identify interactions. The nature of our platform being that these are full-length proteins and we express them in live cells means that it's very adaptable for these types of cell binding or functional assays. One really great feature about the MPA is that we have developed it to be used in 2 different cell lines. So our standard cell line is HEC293 cells. This makes sense, they are a human cell line, and so of course you want to express a human protein array in a human cell line. However, there are instances where the HEC293 cells may not be ideal for the screening of your antibody. For example, B7H3, a target that is quite popular for cancer therapies, is a protein that is highly expressed on HEC cells. So as you can see on the left-hand side here, if you were to screen a B7H3 antibody on the MPA expressed in HEC cells, you're going to get a lot of background binding, which makes it very difficult to distinguish the B7H3 interaction. Because of these sorts of instances, we developed the MPA expression in QT6 cells, which are quail cells, because this gives us an alternative option for screening these types of antibodies. These QT6 cells are also nice to work with, easy to transfect, but since they're avian, they are divergent enough from the HEC cells that it really helps bring down the background binding levels. So in this B7H3 example, you can now see on the right-hand side that screening this antibody on QT6 cells really brought down that background binding, and now you can very nicely distinguish the binding to B7H3. So this is a really nice feature of working with Integral Molecular's membrane proteome array, because for each antibody, we will assess which cell line is ideal. And if you do have a case such as this, it's nice to know that we do have a backup option that we can pivot to using the QT6 cells to be able to move forward with your project and check the specificity, as opposed to having to just cancel the project after we see this type of background level binding, which makes it impossible to analyze the data. Perhaps the most important feature of the membrane proteome array is the fact that we perform all of our screens on unfixed cells. We know that fixative reagents can mask epitopes, which really causes you to run the risk of missing an important interaction. Shown here are 2 head-to-head experiments where we tested a CD19 antibody on the MPA, first on fixed cells, which is our standard protocol. This is shown on the left-hand side. And in this study, we identified binding to an off-target protein, so an immunomodulatory protein that you would definitely want to know about before this antibody goes into humans. However, when we tested the same antibody on the MPA, but this time fixing the cells first, this off-target protein that we had identified and validated in the unfixed experiment was consistently down here in the noise on the fixed cell experiment. So something about the fixative reagent disrupted the epitope of this off-target protein, and so it appears as a false negative and was not recognized in the fixed cell screen. So this really goes to show the importance of performing these types of specificity studies in an unfixed environment that most closely mimics what is seen in the body, so that you are really doing the most comprehensive assessment of your antibody's binding profile. Now you may be thinking to yourself that the MPA platform is all well and good, but will it be accepted by the FDA for IND filing? I won't go into too much detail about the FDA guidance here, as I know our next speaker, Tim McLaughlin, will cover this in more detail. But I will speak generally that specificity testing is required by the FDA, and while tissue cross-reactivity studies have been a standard, newer technologies such as the MPA are being employed and have been accepted by the FDA as part of IND packages. About half of our customers anticipate using MPA data in their IND filings, and some have already successfully done so and are currently in clinical trials. One of these examples is Abound Bio. Abound developed a DSG3 CAR-T therapy for the treatment of mucosal pemphigus vulgaris, which causes painful mucosal blisters. These blisters are caused by autoantibodies against desmoglein 3. So the DSG3 CAR T therapy eliminates these autoantibodies by targeting the corresponding pool of antigen-specific B cells. For this type of therapeutic, mistargeting can kill healthy cells, so it was very important to check the specificity. Cabela did this with the membrane proteome array, and the MPA showed strong on-target and minimal off-target binding. Further investigation showed that the off-target interaction was minimal, and this expression did not trigger the DSG3 CAR T activity, and so this alleviated any cross-reactivity concerns. Cabela did submit the MPA data to the FDA without any tissue cross-reactivity studies and successfully opened IND. They are currently in their phase 1 clinical trial, and I will say that this information, this data, and all of their preclinical work is published in the Journal of Clinical Investigations, so feel free to check that out if you'd want to learn more about their therapeutic. I do want to briefly go over what a standard workflow looks like if you decide to do a project with us. Once we receive your antibodies, each antibody will be subjected to an assay setup screen, a membrane proteome array screen, and then followed by a hit validation. This validation is a 4-point titration testing the binding of the antibody to any hits that were identified in the MPA screen. And we're really looking to make sure that there is a specific interaction indicated by a dose response, which really helps us rule out any false positives from the screen. The deliverable is a report, which will come to you about 4 to 6 weeks after we start the project, and there are some additional options that you can add on at the end of the project, depending on the outcome of your results. To finish out my portion of the webinar, I just want to briefly summarize the things we discussed about what makes the MPA so successful. From having the largest collection of proteins at 6,000 total proteins, to performing everything on unflexed cells by flow cytometry and having 2 different cell lines. These things all really help ensure native protein conformations and preventing false positives and negatives. We do offer quick turnaround times, resulting in an IND-ready report that is accepted by the FDA, and we can screen all different types of biotherapeutic modalities and have done so for over 1,000 molecules. You're welcome to work with Integral Molecular directly, or you can also access our services through our partners shown here, Fairjourney Biologics, Funakoshi, Joint Laboratories, and Huayan, and we are also available on the Science Exchange and Scientist.com platforms. With that, I would like to thank you all so much for listening to my presentation today. Here's my contact information for if you would like to get in touch to discuss anything further. And I would now like to pass it off to our next speaker.
Thank you, Rachel. I'm Tim McLaughlin. I'm from Novartis, working preclinical safety. I'm going to talk to you a little bit today about a survey that we did with a consortia of other biopharmaceutical companies to understand what technologies that they've been using to understand specificity of their antibody therapeutics. Now, the history of the technologies that we use for this come from guidance documents from the FDA and from ICH, originating in 1997, that advised sponsors to take antibodies and pan them across tissue sections, 32 human tissues from 3 donors, and evaluate where your antibody is binding by immunohistochemistry techniques. This will tell you where your antibody binds, but it won't tell you what your antibody binds. Interestingly, in both of these documents, they open the door, really, for the opportunity for sponsors to utilize other techniques than immunohistochemistry against tissues as they become available and validated. So the question really is, what are these alternatives? And over the last several years, these have evolved to be in array format. Originally starting with slides that have random peptide libraries spotted on them and then panning the antibody against this with a fluorophore to see where it might bind to. More recently, this has allowed for the spotting of human GST-tagged proteins that are produced in yeast or bacteria to be spotted onto these slides. And the coverage is fairly decent from a human membrane proteome or even intracellular proteins. This allows evaluation of the binding of the antibody against a fully folded protein. Most recently, This has allowed an understanding of the binding of the antibody in the cellular context. One approach might be to spot plasmids that have a library of human membrane proteins spotted across a slide, and then growing cells over those plasmids, and then panning the antibody again across the slide. An alternative way of doing this is by transfecting cells in a suspended format, and then panning again the antibody across those cells, and then detecting binding by a flow cytometry method. This is a really valuable way to understand specificity, in that it can tell you exactly what your protein binds to. Now, it will not tell you in what tissue and cellular context it would bind to. So there certainly are some positives and negatives to either approach. The tissue cross-reactivity approach utilizes immunohistochemistry, so it's dependent on the antibody being a good IHC reagent, which is actually quite rare for therapeutic antibodies. The IHC methods may not actually fully expose all proteins, so the coverage of the full human membrane proteome might not actually be true in the case of tissue cross-reactivity. There have been some cases of false positive staining, and if you do get staining, it's unclear if that's actually your target protein in an unexpected location or if it's a true cross-reactant. Now, with the arrays, we know for a fact that it's not the full, complete human proteome, at least not at this stage yet. Perhaps someday it would be. And we know that the protein is outside of its normal environments. We're utilizing workhorse cell lines to do this. Some sponsors have done quite a bit of this work, but although some have not utilized this technology at this point in time. And as such, because of that lack of familiarity and experience, some consider this as not validated. So really needs to generate a little bit more widespread experience with this. So based on this, a group of us who focus on preclinical safety within the Biotechnology Innovation Organization wanted to survey industry to see what kind of technologies that they've been using. We use this as a launching pad a paper that we had published on the tissue cross-reactivity study about 10 years ago. And we wanted to ask what sponsors' experience with that assay has been since then. And then since then, these alternatives, these array methodologies have been evolving. We wanted to know if sponsors have been using these, if they've been using them for early screening, if they have been, what kind of platforms that they've been using them with, and what their experiences have been. So briefly, some of the overview of the results from this survey that has now been published. We asked about sponsors' experience with the tissue cross-reactivity study, and there were 26 respondents, and most of the respondents had done up to 30 to 40 different tissue cross-reactivity studies over the last 10 years. They don't do them frequently in non-clinical species. This is not required. Only human tissue cross-reactivity is required, although a small fraction of these sponsors do them to try to validate some of their non-clinical species. They use tool antibodies frequently and the target localization early in the programs. And they have filed some INDs without these TCR studies, or they're considering in the future. And it has always been with oncology INDs. Most are running the tissue cross-reactivity study alongside the GLP tox with GLP tox material. And in the majority of sponsors' experiences with this, the tissue cross-reactivity data has not predicted a safety finding in the preclinical studies. It has not led to, the results of this has not led to a halt in development in a candidate. It hasn't affected the overall preclinical program, hasn't led to a change in clinical trial design, and hasn't led to a clinical hold by regulators. That being said, there are individual examples and anecdotes where the assay was valuable, suggesting that there's still value in performing this assessment to catch that rare, unexpected event. Now, have sponsors started to use alternatives to the tissue cross, namely arrays? And approximately half of the 26 have begun to use arrays for cross-reactivity screening. And the majority of these have not found that they are generating overtly uninterpretable data sets. They report mostly clean or rare false positive data sets that they deem to be quite valuable in understanding the specificity of their antibody. Question was posed in discussion with some of these sponsors is if there is an opportunity to replace the tissue cross-reactivity study with these new technologies. It was noted that clearly, as I pointed out earlier, that the data sets provide information different from what is generated from the TCR, which is the what within the arrays versus the where with the tissue cross. Certainly, some have suggested, well, we could just combine this with a tissue cross assessment, which some do. Certainly, we at Novartis, for non-oncology indications, are currently combining this with the tissue cross. But looking long-term, this does not necessarily seem to be financially feasible in the long term. And we're looking for opportunities to potentially replace with more experience with these array technologies. So bottom line is that there is growing experience with these alternative methods. But we have discovered through discussing the results of this survey with our health authority colleagues at the FDA that the regulatory agencies need to seize more of this data, even if it's just exploratory, because they need to understand what the variability is, what the value of the data that they're getting out of it is. Only half of the respondents that have been performing these note that the data has been sent to health authorities, and we understand that there haven't received any concerning health authority feedback on any of these. We do know that there are some sponsors that are just resistant to replace. They've had over a decade, more than a decade of experience with tissue cross-reactivity as the means to understand specificity, and they haven't had any hiccups with this, that it's just fine. The alternative methods have their own problems. We don't have experience with it, so they want to stick with it. So I think to each their own, really. What we're advocating really is that sponsors have a choice in what technology that they use. And we are long-term would hope that health authorities would accept whatever technology a sponsor would choose to generate this kind of data. So combining all of the information we got out of the survey is that we pretty much are where we were 10 years ago when we published our original paper with the tissue cross-reactivity in 2010. It's not often generating critical safety information. It's not identifying a critical safety issue. However, there's that rare circumstance, perhaps 1 in 10 years, where it did find something that needed to be followed up on. And that there's growing experience with these alternative methods, that we're not quite ready to completely replace it for non-oncology indications, but we should monitor over time and see what sponsors' experiences are. We're really not interested in just lumping this on to existing data, because while we do believe them to be complementary, we don't necessarily believe one to be we actually believe one to be sufficient for understanding the information that we want to gain from this. The regulatory agencies need to see more of this data, and there's some, as I mentioned, some resistance to replace. So engagement with health authorities for the use of these methods is critical. And we have hypothesized that there are a variety of different potential scenarios that sponsors could consider in submitting cross-reactivity data to health authorities. So we could have an array screen coupled with a standard full TCR assay. And as I mentioned, this could be quite expensive, and we're not exactly sure how often that would actually yield more valuable data than just one standard TCR or one array screen alone. We think that it's quite possible that a combination of an array screen coupled with data from target localization using a tool antibody, tissue IHC, would be the best case scenario, and is often done in the early evaluation of a target of antibody developers to understand specifically where their target is going to be and where the tissues that they're going to want to monitor are. And this is much easier done in a research non-GLP setting with an antibody that is optimized for immunohistochemistry, not necessarily the one that is optimized for therapeutic use. So with that, I want to thank all of the people that were involved with the generation of the survey across several different companies. And with that, I believe that we'll be transitioning to our final speaker.
Thank you, Tim. I am Dimitar Dimitrov, Chief Scientific Officer of Abound Bio, Director of Center Antibody-Therapeutics. First of all, I would like to thank the organizers for inviting me. I'll talk initially about our company and our Center of Antibody-Therapeutics at the university, then about our platform features, display, and some selected antibodies. And then I'll tell you examples of how membrane proteome arrays helped us to characterize our antibodies against SARS-CoV-2 and also some antibodies against cancer. The Center for Antibody Therapeutics was founded by John Mellors and myself 4 years ago, and it's in the University of Pittsburgh School of Medicine. Its most important mission is to develop and commercialize antibody-based therapeutics to address unmet medical needs, and as we use phage in this display, human antibodies, collaborations with university professors, scientists, and also outside, also training and spin-off companies. We discover new antibodies, and also we can make antibody-drug conjugates, CARs, IgGs, bispecific, bias, trispecific, antibody-based fusion proteins. Abound Bio was started 2 years ago with the major goal to discover, develop, and commercialize antibodies for unmet medical needs, including lethal cancers and infectious diseases. The two founders are Dr. Mellors, who is a distinguished professor at Pitt. Also, he is co-founder of another company, ID Connect, and myself. We started with a $2 million in seed capital, specialized in discovering potent, specific, fully human antibodies in various formats to difficult targets. Also, we have very large libraries, trillion specificity, and use proprietary methods for rapid isolation and engineering of those antibodies. Has already signed several partnerships and licensing agreements. We display our libraries displayed on phage list as I already said, and then we do panning or sorting, and then selected binders we can engineer in various formats. While I was at NIH, we developed a number of antibodies. One of them was approved for clinical use on compassionate basis in Australia. Another one was licensed for a large amount of money to Juno, and also trial antibodies, especially the last one against mesothelin to Atara, and then sub-licensed to Bayer for $610 million total deal. As we all know, specificity is extremely important for safety. Actually, FDA asks for safety and then efficacy. And there are different ways, of course, to evaluate the specificity of our antibodies, but why membrane protein arrays? Why we selected that platform? First, it really has many, virtually all important 6,000 membrane proteins. Also, the cells they use are unfixed, so they preserve their native conformation, which is very important, high quality, and then very quick. Actually, our experience shows that, and I knew that it's like within 4 weeks, or sometimes even quicker, especially like for our SARS-2 antibodies. 4 weeks is really quick turnover. And also there is validation by flow cytometry, and I had a very good previous experience with this well-housed NH. We evaluated a number of antibodies against SARS-CoV-2 and also against various cancer-related proteins. As you can see here, all these 5 antibodies against SARS-CoV-2 were evaluated, and there were no off-target binding for all those antibodies, and they're also in 2 different formats, as IgG1 and also as a fusion protein of with Fc, VHR or VH domains. We also evaluated a number of cancer-related proteins, antibodies against those proteins, and 2 of them are against PSMA. And actually, in this case, we found, and that's really very interesting, I'll show you later, that it also bound to another protein. All our other antibodies, one deny against CD66 or SICAM5, also 3 antibodies against mesothelin, none, and then 2 other antibodies against CD2726. There are no off-target bindings, which we're really very happy about. I'll show you 2 examples, SARS-CoV-2 antibodies, AB1 and AB8. These are the data for our antibody against SARS-CoV-2, the domain one, which was published last year. And we can see the data here, like the target binding and then the membrane protein area, and there is a positive control in SARS-CoV-2, and it binds to SARS-CoV-2, but doesn't bind to anything else. So the question, again, as I mentioned, it's very important that we do so-called de-risking for safety in the environmental maps, and there are some examples of monoclonal antibodies cross-reactive with unrelated proteins. I remember in the past there are some antibodies against to HAV1, GP41, and a number of viruses, they use various strategies in order to bind, to redirect the antibodies to human proteins. And also, there is some evidence that COVID patients generate self-reactive antibodies, which may lead to autoimmune symptoms. So the membrane protein array was used to profile our, as I already mentioned, high affinity antibody domain that can block the AC2 interaction. And this is part of Abound Bio's COVID-19 therapeutic program. We also tested another antibody, IgAB1, which actually was produced, GMP produced, and ready for clinical trial. And as you can see here, it really doesn't bind to any of the human proteins, except some weak binding as expected to FcR receptor 1, Fc gamma receptor 1, which was, of course, made us very, very happy. Several examples for our antibodies against cancer-related proteins. I just will show you 2 of them, to mesothelin and PSMA. Here you can see 3 different antibody domains, they're all domains targeting mesothelin. And you can see that they really don't bind to any human proteins except some, again, Fc gamma receptors, but here's mesothelin, and they're all the same. So there is no off-target binding for all of those 3 antibodies, which are now undergoing animal testing. Here is the example I mentioned earlier, that there are 2 VHs which really bind not only to the target PSMA, but also to another protein, TMEM178A. And they're 2 different, but not so much different, but they both are binding to this additional protein. So in this case, what we do is we really actually, this is what we're doing right now. We are testing to see which cells express this protein. We see also in the literature what are the data, but we are doing also our own experiments in order to evaluate potential toxicity. So in case we find that these are really important cells and it's really important, then we can engineer the antibodies to eliminate this binding, and we have this kind of techniques. Or alternatively, if it's not possible for whatever reason, then we can really just make another panning and identify different binders. But the major point is that this is really very, very useful. We don't want to develop the antibody and all these steps and finally to find in humans that it's toxic. So this is really very important. So in summary, we found that the membrane protein assay is very fast, reproducible, and reliable for evaluation of our antibody specificity. And those antibodies against SARS-CoV-2 and cancer-related proteins were highly specific, except 2 antibodies which bound to another protein. But this is really very important for us that if the antibody binds to another protein, then we can find those cells which express that protein and see how important is this. And if those cells are important, then we can engineer the antibody to eliminate binding to that protein, or just we can select different antibodies to the same target. So in conclusion, we plan to continue to use the assay for in vitro testing of our antibody specificity. So many people contributed to the data to discovery characterization of our antibodies. I just will mention Wei Li and Zihua San and Chen Chuan and Don Trujillo, who really, and Dusan Baik, who really contributed most of the work. So for the sake of time, I'm not going to acknowledge the others, but as you can see also, our company, Martin Nuro, Eric Peterson, Alex Conner, they all contributed to this. Finally, the funding from UPMC, as you can see, if you go to Pittsburgh, beautiful Pittsburgh, the highest building in downtown is UPMC, and also National Cancer Institutes and Baldwin Foundation. And this ends my talk. I hope you enjoy. And now I give back the microphone to the moderator. Thank you very much. Bye.
Thank you very much to all three of our presenters. We do have some time now for a question and answer period. I have some questions that have already come in. Please feel free to chat your questions to all panelists. Our first question is for Rachel. Can the MPA be used to identify the receptor for my protein of interest?
Thanks, Elizabeth. Yes, that's a good question. So today we focus a lot on the specificity testing application of the membrane proteome array, but as I'm sure many of you can deduce, this platform is also amenable to doing what we refer to as deorphaning or receptor identification. And we do perform a lot of those types of projects as well. It's basically the same format in terms of the technology and what we do. It's just answering a different sort of question. So yes, the orphaning or receptor identification work can definitely be performed on the MPA.
Perfect, thank you. A question for Tim. You showed in your slides that TCR data has rarely led to a change in the clinical program. Is this because there are very few off-targets found by TCR, or that the off-targets found are not relevant?
It's a very good question. It's tough to tell, really. I mean, I think sort of alluding to another question relating if how often are we missing something in a TCR that ends up becoming a problem in the clinic. I can tell you that I'm familiar with one circumstance, I believe, where there was a VEGF antibody that did have an adverse reaction in the clinic that was ended up finding that it did cross-react to something else that led to that. But I think that that's a very rare circumstance, and I'm unfamiliar with anything else. I think at the end of the day, there's the potential for an antibody to be binding something else, but not causing anything dramatic in the clinic, at least initially. I think that at the end of the day, what you're looking for is a full understanding of what kind of pharmacology your antibody is imparting, whether it be for efficacy or safety. And then if there is a cross-reactivity, understanding whether or not it was consequential by evaluating that in a non-clinical sense, or if maybe it might be helpful from an efficacy sort of sense. So it's really just about understanding the full component of pharmacology that's imparted by your antibody. I hope that answers the question.
All right. Thank you so much. Our next question is for Dimitar. Do you consider, or did you consider, using TCR for your programs? Why did you pick this approach instead?
Oh, because our program is very initial stage, and TCR is much more expensive. So we can use also TCR later if we have some problems with the membrane proteome array. So this is why we started with that, and also because we can have like multiple antibodies in general, because our company is really a company we try to save money.
Excellent reason. Going
Yeah.
back to Rachel, how often has MPA been used to submit an IND, and have applications been filed only to the FDA using MPA data for specificity, or also to other regulatory agencies like EMA and MPA and so forth?
Yeah, so like I mentioned on one of my slides, we do know in our own surveying of our customers, we do know that about half of them do plan to use the MPA in their IND applications. We do also know of customers who have successfully done so, Capilleta being one of them. There are others that I can't be as public about, obviously. We are reliant on customers kind of coming back and letting us know, since these things usually are not public. But yes, we do know of about a third of the customers who have filed either with or without TCR studies. And we know that these filings have been done with the FDA, but then also with other regulatory agents overseas in Asia and in Europe as well.
Thank you. And this is also about regulatory agencies. This one's for you, Tim. Do you know of any cases where off-target reactivity was found by TCR and a regulatory agency asked for additional information about the identity of the off-target?
Yeah, great question, and the answer is yes. It doesn't happen that frequently, but when it does happen, it can definitely send sponsors down a rabbit hole. We actually outlined a number of these case studies in our 2010 paper, where sponsors were forced to do a lot of follow-up work based on some kind of staining, whether it be real or not, in the TCR. And to be honest with you, that's kind of why we initiated this survey in the first place regarding alternatives, because we feel that these array screens, while they don't tell you where, this what question of what your antibody is binding to seems to be much, much more valuable than the where sometimes. Yes, I think that this data of the what could potentially really mitigate some of these regulatory concerns when they crop up.
Thank you. Our next question for Dimitar. How important do you think it is to test antibodies targeted for viral proteins for specificity compared to testing antibodies targeted at human proteins? Have you had any personal experience with off-targets for your SARS-CoV-2 antibodies?
No, we didn't have any. So I think for both viruses and cancer, it's very important to, especially if you go to put them in humans, it's very important. So we will like test always with the membrane proteome arrays for both viruses and cancers. For SARS-CoV-2, yeah, we tested many antibodies, at least, I showed you 4, and we never found any cross-reactivity. It seems that in contrast to HIV, SARS-CoV-2 is not kind of causing many autoimmune antibodies. So it has not been kind of adapted as much as HAV for mimicking human proteins and this way escaping immune responses. Did I answer the question?
Well, if it didn't, if the person who chatted it in could clarify in the chat panel, that would be great.
Mm-hmm.
Going back to you, Rachel. Okay. How do you ensure proteins are expressed?
A very reasonable question when you're thinking about this many proteins in an array. So first, I want to just give a little bit of the background, like I said in the beginning, about integral molecular and what we do. So we are a company that was built on working with membrane proteins, and we've done that for 20 years. So we know all the tips and tricks and have many tools in our toolbox for expressing membrane proteins very well. For the MPA specifically, we do have tags on about 90% of the array, and that has been particularly helpful for us to assess the expression of each protein. And in the development of the MPA, this tag was particularly useful because we could then see what was not expressing well and go back in and do some tooling around with the tips and tricks that we know and what we have in our toolbox to fix those proteins and be able to, then offer an improved and well-validated MPA. So yes, that's a very good question. It comes up a lot. It is tough with 6,000 different proteins that will all express a little bit differently, of course, but with the tag, it really gives us a good big picture.
Excellent. Thank you. Another question for you, Tim. The survey you discussed was conducted up till 2018. Can you tell us a bit more about how the use of cell arrays has increased or expanded since then?
Yeah, I mean, just really anecdotally, my understanding is certainly has increased. I think even just doing the survey sort of inspired some discussion in some of the companies to say, well, hey, it appears that this is really gaining traction. Why don't we do some more of this, continue to pressure test this technology internally? And I know for a fact a few companies that hadn't been doing them before are now starting to implement them as more routine analyses as well. So certainly we'll keep tabs on this over the course of time and see how much it might increase. Maybe 2-3 years from now have another check-in, but we have anecdotally heard more increases in uptake.
Thank you. A question from Diana for you, Tim. How frequently does a TCR miss an off-target that causes issues later, for example, in clinical trials?
Yeah, I think sort of referring a similar question that was asked before. We're not seeing very often antibodies go into clinical trials and clearly have some safety event that does not appear to be at all related to pharmacology or typical antibody administration into humans. Now and again, there was actually a publication a little while back of an anti-VEGF antibody that saw that and then went and did an array and found that it actually had an off-target that seemed to imply it was the culprit for the safety issue. So like I said before, I think that it's more about doing these things to have a full understanding of what the biology of your product is. And now we have a technology to get that more information, whereas just doing IHC on tissues it gave you some information, but really didn't give you much about the characteristics of the binding. So.
Perfect. Thank you. This question is for you, Rachel, from David. Is it also possible to screen non-antibody binders, for example, novel protein scaffolds, that may be as small as 10 kilodaltons in a comparative analysis to MAb controls?
Yes, thanks, David. Yes, we can screen non-antibody molecules on the membrane proteome array. The most important thing to keep in mind is whether there is a method to detect that particular molecule by flow cytometry. Since that is our detection platform, there just has to be some way, whether if it's directly conjugated with a fluorophore, or if it can be biotinylated or fused to an FC. Those all will then allow us to perform the detection by flow cytometry. We have worked with a lot of non-standard molecules before in the past, and are usually able to find some way that we can work with the customer's molecule modality, whatever it is that they have that they're trying to screen. So if that's something of interest to you, please get in touch with us, and myself and our scientists are happy to speak with you more about the type of molecule you have and what some options might be for screening it.
All right, thank you. We're getting close to the top of the hour. We have time for just one more question. This one also for you, Rachel. Is the membrane proteome expressed in the avian cells published or deposited somewhere as a reference?
That's a really good question. We do have a number of publications that show the MPA data. Off the top of my head, I can't remember, to be honest, which ones have HEC data, which ones have the avian cell data. But it's something that we can certainly look into and get back to you directly with an email.
All right, thank you. We do have quite a few more questions. I will send those along to our presenters, and you'll receive those answers via email. I would like to very much thank our presenters today, Dmitry Dimitrov, Rachel Fong, and Tim McLaughlin. I'd like to thank Integral Molecular for sponsoring today's web symposium. Finally, I'd like to thank all of you who came and spent this time with us. We're very grateful you came. We know things are crazy out there, and we're glad that you had some time to share with us. So on behalf of Cambridge Health Tech Institute's Global Web Symposia series, thank you all so very much, and have a great rest of your day.